Hello, my name is Hillary Schiff, and today I'm going to be telling you a little bit about some of my work and research that I have done in the Boitano lab as an undergraduate researcher. This project that I'm going to be talking about is a project I worked on in lab, and the focus of this project was to develop new, more physiologically relevant screening protocols for potential biased antagonists of PAR2. The overall purpose is to help in allergic asthma drug development. So getting into a little bit of background here, and I'm going to zoom in on this figure. PAR2, or protease activated receptor 2, is a type of G protein coupled receptor that's found in the airway. And PAR2 actually has a paradoxical association with allergic asthma, meaning that it both contributes to and helps relieve symptoms. And how that works is PAR2 is associated with two downstream signaling pathways. This G protein coupled pathway causes bronchorelaxation and is protective of the airway. And then this beta arrestin coupled pathway is inflammatory and contributes to those symptoms. So you may be thinking, if we want to design an ideal treatment for allergic asthma that targets PAR2, we would want to selectively block this beta arrestin inflammatory pathway to minimize allergic asthma symptoms. And that is called a biased antagonist. Now, in collaboration with Dr. Joseph Wagner's lab, we believe we have just that. And this compound is called C781. And these two figures, collected by some of my colleagues in lab, simply target downstream characteristic signaling pathways to demonstrate that C781 is selectively blocking the beta arrestin pathway activation. You can see that here. So a big focus of my project moving forward from that is that this data was collected using an artificial agonist called 2AT or C134. And since this data looks really promising, we want to move forward in characterizing C781's blocking ability using naturally occurring enzymes that we may find in the airway. And so those two enzymes that I decided to use were neutrophil elastase and trypsin. So the first part of my project was to characterize neutrophil elastase and trypsin activation of PAR2. And in order to do that, I used a technique called the RTCA, and it's the real-time cell analyzer. And how this works is you plate cells on a plate with gold electrodes at the bottom of each well. And when you put that plate in the machine, it puts a current through the gold electrodes and measures impedance in a unit called cell index. So there's several things that can cause impeded electron flow, but the specific thing we were looking for was impeded electron flow due to morphological changes to the cell due to activation of the receptor, or morphological changes at the receptor due to receptor activation. So what this looks like is it looks like a really steep increase in cell index, and you'll see that as we look at the figures here. So what I did at first to establish that these proteases could be used to model PAR2 activation is I treated cells with various concentrations of these figures up here are using trypsin, and these down here are using elastase. And then what I did is I did that exact same treatment on cells that had the gene for PAR2 knocked out. So cells that were not expressing PAR2 at all. And this was in order to show that our enzymes were acting as specific targets on PAR2, the receptor of interest. So what we can see here with trypsin was very specific, and elastase was sufficiently specific, but you can see some non-specific effects on these cells without PAR2 after treatment. And then these experiments enabled me to accumulate some data and really characterize which concentrations of trypsin were causing which level of activation of PAR2. And the same for neutrophil elastase later on. So then using that information, my second goal of this experiment was to characterize the block of C781. And I did this using the EC50 and the EC80, so concentrations that activated PAR2 to a certain extent. And I treated those with 10 micromolars and 6 micromolar of C781 respectively. And as you can see here, this is the effect without any C781 treatment. And you can see that with that C781 treatment, you do get an attenuated response. And the same for neutrophil elastase. So this is really promising moving forward. It showed that we can model C781's blocking ability on PAR2 as activated by naturally occurring enzymes that would be in the body. And so moving forward, this is not really sufficient to demonstrate biased antagonism. In order to do that, we would really need to use fluorescent-based signaling assays to target those downstream signaling pathways specifically. And then also this is an in vitro characterization. So moving forward in characterizing C781 as a potential drug candidate, we will want to move into in vivo screenings as well. So thank you very much, and yeah, have a great day.